Welcome to one of the most fascinating and crucial topics in molecular biology. Imagine you are managing a massive, high-tech factory that builds luxury cars from thousands of tiny, highly specific parts. If a worker accidentally installs a fragile bicycle tire instead of a high-performance car tire, the entire vehicle becomes useless and incredibly dangerous. In your body, the factory is the cell, the assembly line is the ribosome, and the luxury cars are the proteins that keep you alive. Today, we are going to explore the fidelity of protein synthesis, which is the strict, multi-layered quality control system that prevents catastrophic biological crashes. Now that we have our factory analogy in mind, let us look at what is actually happening inside your cells. Your DNA dictates the exact, perfect order of 20 different amino acids to build specific proteins. The fidelity of protein synthesis refers to the extreme accuracy with which the genetic code in messenger RNA is translated into a specific polypeptide sequence. This is not just a single step, but a highly coordinated series of molecular checkpoints. The cell spends an enormous amount of energy double-checking its work because these proteins are the molecular machines that do almost everything in your body, from digesting food to firing neurons in your brain. Understanding that fidelity is a multi-step process brings us to the specific checkpoints the cell uses. The two primary checkpoints occur first during the charging of transfer RNA by enzymes called amino-acyl tRNA synthetases, and second during the matching of the messenger RNA codon to the transfer RNA anticodon inside the ribosome. Through brilliant evolutionary mechanisms like the double sieve editing process and kinetic proofreading, the cell achieves an astonishingly low error rate. Typically, the translation machinery makes only one mistake for every 10,000 to 100,000 amino acids it links together. This precision is absolutely vital for survival. This incredible precision naturally leads us to ask, what happens when the system fails? If the fidelity of protein synthesis drops, the cell starts producing misfolded, non-functional, or even toxic proteins. These defective proteins clump together and destroy cellular structures. In real life, a failure in this quality control system leads to severe neurodegenerative diseases, such as Charcot-Marie-Tooth disease, where nerve cells slowly die off due to toxic protein buildup. Without this rigorous fidelity, life as we know it would simply be impossible, as our cells would drown in a sea of defective, garbage proteins. To truly appreciate how impressive this biological accuracy is, we can look at it through the lens of probability. When a ribosome builds a protein, it must make a correct choice at every single position along the growing chain. This relationship is beautifully captured by a simple probability formula. P represents the overall probability of synthesizing a completely error-free protein. Epsilon represents the error rate per codon, which is the tiny fraction of times the ribosome inserts the wrong amino acid. N represents the total number of amino acids in the protein chain. The term 1 minus epsilon is the probability of getting exactly one amino acid correct. By raising this to the power of n, we multiply the probability of success for every single step. If the error rate increases, or if the protein length increases, the overall probability of getting a perfect protein drops significantly. Now that we have our probability formula, let us see this with a concrete example. Let us imagine a human cell is synthesizing a medium-sized protein that is exactly 1,000 amino acids long. The cellular quality control mechanisms maintain an average error rate of one mistake per 10,000 amino acids added, so epsilon is 0.0001. We want to find the probability that the entire synthesized protein is completely free of any errors. We substitute our values into the formula, replacing epsilon and n. Subtracting the error rate from 1 gives us 0.9999, meaning there is a 99.99% chance that any individual amino acid is placed correctly. Raising this to the power of 1,000 gives us approximately 0.9048. This means that over 90% of the synthesized proteins will be absolutely perfect, proving how robust this system is. Having seen the mathematical probability of success, we must now ask how the cell physically achieves this incredibly low error rate. The answer lies in the laws of thermodynamics and energy differences. When the correct transfer RNA binds to the ribosome, it forms perfect hydrogen bonds. In contrast, an incorrect transfer RNA forms weaker, mismatched bonds. This difference in binding strength translates directly to a difference in free energy, which dictates how easily the cell can discriminate between the right and wrong molecules. Thermodynamic proofreading is the fundamental physical mechanism that makes life possible at a molecular level. This concept of free energy differences brings us to a fundamental thermodynamic equation. We can calculate the exact energy difference required to achieve a specific error rate. Delta delta G represents the difference in binding free energy between the correct and incorrect transfer RNA molecules, measured in joules per mole. R is the universal gas constant, and T is the absolute temperature in Kelvin, which is typically around 310 Kelvin for the human body. E represents the error fraction. The natural logarithm function here shows a crucial relationship. To make the error rate exponentially smaller, the cell must linearly increase the energy difference between the correct and incorrect molecules. If the temperature increases, the required energy difference also increases to overcome thermal noise. Now that we have used this thermodynamic formula, let us apply it to a real biological scenario. Suppose an aminoacyl tRNA synthetase enzyme needs to distinguish between two very similar amino acids, valine and isoleucine, to maintain an error rate of 0.0001. We know the gas constant, and we assume a human body temperature of 310 Kelvin. We plug these values into our equation to find the exact energy gap the enzyme must create. Multiplying the gas constant and temperature gives us the thermal energy in the environment that the enzyme must overcome. The natural logarithm of our error rate is negative 9.21. The negative signs cancel out, giving us a final required energy difference of approximately 23,738 joules per mole, or 23.7 kilojoules per mole. This calculated energy value of 23.7 kilojoules per mole leads us to an important physical limitation. This result tells us that the enzyme must bind the correct amino acid with significantly more energy than the incorrect one. However, a single hydrogen bond only provides about 4 to 20 kilojoules per mole of energy. Because the difference between similar amino acids might only be a single methyl group, a simple binding pocket is often not enough to reach this high energy difference. The thermal noise of the environment is simply too great. Therefore, the enzyme can't rely on initial binding alone. It has to use a secondary editing site to double-check its work and physically remove mistakes. To visualize how the cell overcomes this energy limitation, let us look at the double sieve editing mechanism. In this diagram, you're seeing the aminoacyl tRNA synthetase enzyme, represented by the large blue shape. It has two distinct pockets, the activation site, or CIV1, and the editing site, or CIV2. Notice how the activation site completely blocks larger incorrect amino acids from entering. That is the first CIV. However, smaller incorrect amino acids, shown in red, can sometimes slip in. This is where the second CIV comes into play. The smaller incorrect amino acid is shifted over to the editing site. Because it fits perfectly into this second pocket, it is cleaved and hydrolyzed, effectively destroying the mistake before it can be attached to the transfer RNA. The correct amino acid, shown in green, does not fit into the editing site and safely proceeds to bind with the transfer RNA. We have covered the full topic of protein synthesis fidelity, so let us reinforce the most important ideas. First, fidelity is not a luxury. It is a magnificent, multi-layered quality control system that keeps our cells functioning and prevents severe diseases. Second, we saw mathematically that the probability of building a perfect protein depends heavily on maintaining an incredibly low error rate across the entire length of the chain. Third, we learned that achieving this accuracy requires significant differences in binding free energy to overcome thermal noise. Finally, because simple binding is not enough, cells utilize highly specific enzymes that act as double sieves to physically proofread and edit mistakes. This delicate balance of thermodynamics and probability ensures that the genetic code is translated flawlessly every single second.